Thank you everyone for this really lovely introduction. Today I am genuinely excited because the program looks great and I think it's going to be a very interesting symposium, but also because this is the first time that I'm going to present this work that is not only my work, but is also the work of several other people here at biocrates, looking into complex chronic diseases through the prism of metabolomics. And this took us to finding very quickly that there's this commonality that is very well studied through metabolomics, and that would really help us, if we took it more into account, that would really help us to better tackle those complex chronic diseases. So without further ado, let me just introduce the structure of this talk, and then we'll look in more detail at what this commonality is. So all this work is condensed into a white paper that I will mention multiple times during the talk. If there's anything interesting to you, if I've piqued your interest, please go on the biocrates website and download the white paper. There will be everything I talked about today and a lot more. But today I will focus on the core hypothesis of the white paper before looking into the three main steps of this core hypothesis. I won't have time to go into the detail of the 6 diseases that we studied in the white paper, but you can get more information on this when you look at the document itself. And I'll finish by discussing how we can measure metabolic imbalance, which in short is the answer to the question, like the common origin to all diseases that we discuss in the white paper, whether it's metabolic diseases like diabetes or liver disease, or diseases like cancer or neurological diseases or autoimmune diseases. At the root of it, you can always find a metabolic imbalance, very often that is brought about by food, by what we eat, by its impact on the microbiome, and how once the balance is tilted, disease ensures. So this is the core idea of the white paper and what I will discuss further in this presentation. I think everybody knows more or less that there are complex chronic diseases that are on the rise globally. So if you take the whole planet as a scale, we know for the last decades that diseases like obesity, type 2 diabetes, but also dementia or different types of cancers are on the rise, and the projections are pretty bleak for the future decades. And because we're today at Metabolomics India, I wanted to also take a look at the situation in India and combining values from different reviews. This is what I found. So obesity is also on the rise, of course, in India, and possibly because Western style diet is perhaps becoming more prevalent in certain parts of the country. And metabolic syndrome was the one that shocked me the most. I didn't expect that some estimates go up to 40% of the population that might be on their way to metabolic syndrome. And diabetes is on the rise as well. So are cancers. Even though the numbers are not as striking, they are increasing every year. And depression is also one that I didn't expect to see. So I chose to talk more today in this presentation about diabetes, and specifically type 1 diabetes and depression. And I will come back to those later in the talk. But what is really also striking with India is that unlike other countries, there are large geographic differences. So there are parts of the country where you will see very high numbers, and others where the population seems not so touched by these diseases. And why do I want to talk about these complex diseases in a symposium about metabolomics? It's because complex diseases by definition cannot be explained by genetics alone. It's the same as multifactorial diseases. There are usually genetic predispositions, but it's not enough to explain why a person will develop the disease later on. And so the metabolome is a beautiful place to look at other influences that are going to contribute to someone developing a disease. The exposome in particular, the way we live our lives, the pathogens that we're exposed to, the medication we take, and very importantly, what we eat. This all contributes to the metabolites that we have in our body. And that food that we eat also contributes to shaping our gut microbiome, that in itself is also a contributor to the metabolome. And together with the effects of our genetics, contributes to the metabolic picture that we have in metabolomics. And that is a very powerful tool to look at complex diseases. So let's go to the core hypothesis of the white paper and this commonality of complex diseases. What happens basically is that we're tilting the balance with food. This is the the hypothesis we went with, because of course a Western style diet, a diet that is characterized by excessive levels of saturated fats and refined sugars, as well as additives like sweeteners and emulsifiers, are associated or risk factors of all of the diseases that I've mentioned previously. And this metabolic imbalance primes the body for disease. But the Western style diet is not only excess, it's also insufficiencies. It's not providing enough fiber, it's not providing enough polyunsaturated fatty acids or vitamins, minerals, and antioxidants. And so this causes an imbalance in nutrient intake, modified levels of metabolites in the blood and in different organs. This is going to cause a weakening of membranes and cellular structure and eventually organ dysfunction. And so the core hypothesis of the white paper is summarized in this figure, where you see 3 steps, starting with the Western style diet, which is a form of malnutrition, even though to some extent it's overnutrition, but it's also undernutrition for certain nutrients. And this, combined with its effect on the gut microbiome, is going to converge onto different levels of metabolites that trigger early stage metabolic disease. That's the second step. And once this is started, progressively it leads to insulin resistance. In adults, we're speaking of adults eating multiple times a day for decades, something that is progressively eroding their system. Insulin resistance becomes installed in the system, and through 3 different paths can lead to complex chronic diseases. And over time, you of course, you won't develop all the diseases, but you usually get 1 or more of those diseases as you get older, unless something is done to prevent it. And those 3 routes are inflammation that can be caused by hyperinsulinemia or other factors that cause systemic inflammation, that is an underlying condition of all of the diseases that are listed here. We also have intracellular malnutrition, for example, because glucose cannot enter cells due to insulin resistance. Also, when short-chain fatty acids are not available because fiber is limited in this diet, and the microbiome also cannot produce those short-chain fatty acids. And this also causes difficulties for the cells that in the end result in disease. And the system tries to reprogram itself. It's very flexible, it's very strong, it's very smart. And it tries to reprogram to compensate for these deficiencies, and often relying on mitochondria to bring up their metabolism, especially using fatty acids as energy source. But I will discuss this in a later slide. Mitochondrial structure and function can be already impaired from the diet, and therefore might not be the mitochondria might not be up to the task to compensate, at least not forever. They do for a while, but they can't do it forever. So let's look at these 3 steps that we have on this picture in more detail. The first step, oops, sorry, I went back. The first step is an imbalance in the gut microbiome. So the Western style diet shapes the gut microbiome. And of course, we know that food acts as a prebiotic, so it's the food of our gut microbiome bacteria, but it's also a disturbance that can influence the different strains of bacteria that live in our intestine. And of course, one of the main effects that is seen of a Western style diet is the loss of alpha diversity. So the richness of the different types of bacteria is lost, and so this healthy microbiome characterized by this diversity of strains is lost. And especially it becomes very problematic if we lose strains that generate short-chain fatty acids. But of course, if we don't provide any fiber, this is like a double way to lead to that problem. The consequences for our organs and our health are going to be primarily local. So a vulnerability of the intestinal lining, the mucus layer is going to get thinner. The bacteria are going to be able to cross the barrier that is formed by epithelial cells in the gut. This is going to cause local inflammation that eventually blooms into low-grade but chronic systemic inflammation. And in the first months of life, this also can lead to faulty innate immune training that can result into autoimmune diseases. This we discuss in detail in the white paper as well. And over decades of exposure through the diet that we eat, this will also promote insulin resistance, this weakening of membranes I mentioned before, and also increased levels of uremic toxins, amongst other things. So we have this imbalance again in the gut microbiome this time, caused by the food. is going to again have an impact on the metabolite levels that we can measure, for example, with metabolomics. And it's going to contribute to systemic dysfunction overall. And here you see on the right, those little symbols. We have a symbol for insulin resistance, and then the 3 branches that I described before, the intracellular malnutrition, mitochondrial issues, and inflammation. And we will see these again when we look in the second step at the imbalances in metabolites that are measured, because these metabolites are the triggers for the early stage metabolic disease through those 3 routes. And so you can link certain metabolites to these 3 different branches of the model. For inflammation, typical cases that we see in metabolomics are the ratio of kynurenine to tryptophan. That is very indicative of inflammation, as well the ratio of lysophosphatidyl-cholines to phosphatidylcholines. For mitochondrial dysfunction, very often we see in blood an increase in acylcarnitines. And this is also indicative of mitochondria that cannot perform beta oxidation properly. And for intracellular malnutrition, it's a bit more difficult because we're talking at the cell level. So with metabolomics, especially blood metabolomics, it can be more difficult to see, but there are signs, and there are other methods also that can help you to monitor this. So in the white paper, we go really in detail for like globally, and also for each disease into which metabolites have been shown to change and what that could mean. So if you're interested in this, please refer to the white paper. I just want to show 3 quick examples to give you a taste a little bit. The first example is an example of a metabolite that's often studied in the context of inflammation. It's arachidonic acid. Maybe you recognize those cards. They're from the Metabolite of the Month blogs that we post every month. So a lot of the metabolites related to complex diseases are already metabolites of the month, so you can learn a lot already there. But of course, so this one is typically released from the metabolism of phospholipids that are in membranes, and it's a precursor of oxylipins or icosanoids, and a ligand for certain receptors as well. It's very active, very powerful, and very well studied in the context of inflammation. Another one that I really want to bring your attention to is deoxycholic acid. It's a secondary bile acid, and it's really discussed at length in the white paper because this one may be an extremely powerful, and it's currently my favorite metabolite, but it may be a very powerful metabolite in the context of complex diseases. As you see from its structure, it's a derivative of cholesterol. And if you know anything about lipid membranes, you know that they're very rich in cholesterol. And secondary bile acids are produced by our gut microbiome. And some of them, like deoxycholic acid, become extremely good at intercalating in lipid membranes. And this becomes very dangerous for the structure of the membrane, especially in mitochondria. And when you know that a lot of the enzymes in mitochondria are lodged in the membranes, you can see the effect this can have on metabolism, from the structural impact to the impact on the metabolism as a secondary event. But also secondary bile acids like DCA are direct ligands of receptors, so they have their direct effect on gene expression and metabolism, and also immune regulation. Very powerful metabolites. The third example I wanted to mention today is butyric acid, or in general short-chain fatty acids, small but mighty fatty acids. They are energy source for certain cells, especially intestinal epithelial cells that rely on them. So the gut microbiome produces them from the fiber we eat, and they're directly available for the epithelial cells in the gut. When they don't have this, there are lots of problems that can happen. But these small molecules also have the power to regulate hormonal signals, including insulin signaling, and they're also ligands for certain receptors. So you see, these metabolites really have an impact, and once they converge onto building up insulin resistance and pushing our metabolism through those 3 routes of inflammation, intracellular malnutrition, and mitochondrial dysfunction, we start to see over time an imbalance in organ health, which is step 3. Unfortunately, in 20 minutes, I don't have time to go through each model, even though I would love to, but these are the metabolic models that we built for 6 diseases. So priming for cancer, Alzheimer's disease, depression, type 1 diabetes, inflammatory bowel disease, and multiple sclerosis, as well as a model of priming the system for autoimmunity due to the Western style diet and its effect on the microbiome. And I will just point your attention to the fact that even just on those pictures, you can see where the balance is shifting. There is always one or more places in the picture where there's a balance between different cell types, between different metabolites that is shifted, and that is going to be one of the causes for the onset of disease. But I want to show you two very interesting examples of the interpretations we could make after building those models. The example of depression I've already discussed before, so you may have heard it before, but it's really relevant in the context of this presentation. So we built our model of the metabolic model of depression, and then we had a couple of studies where antidepressants had been studied with metabolomics. So we could follow which of our metabolites of interest were changed. And in particular, in our model of depression, ADMA, a metabolite derived from arginine, was very high. And we saw that both models compensate for this ADMA, these high levels of ADMA in one way, but neither of them actually reduces ADMA levels. So we suggest really targeting ADMA for new drugs and seeing if this brings relief to people, especially who are not responsive to already existing antidepressants. Another very interesting finding in type 1 diabetes actually originated from a study in multiple sclerosis. So this is the model for multiple sclerosis. I'm not going to get in detail with this, but I just want to discuss a study by a German group from 2020, where they supplemented patients who have multiple sclerosis for 2 weeks, only 2 weeks, with propionic acid, a short-chain fatty acid. And what they saw is an increase in functional regulatory T cells and a decrease in pro-inflammatory T helper cells. So the immune system going back to defending rather than attacking. And after following up the patients 3 years after this 2-week exposure or supplementation, they saw a reduced annual relapse rate in multiple sclerosis, stabilization of the profile of the disease, and reduced brain atrophy, which I think is an amazing result. And all of this from exposing them to a short-chain fatty acid, which in our model is one of the reasons, the decrease of short-chain fatty acids is one of the reasons why the immune system is not properly activated in childhood and therefore primes our system for autoimmunity. So this was really interesting results. But then, sorry, other direction. Then when we put these results in the context of type 1 diabetes, which is also an autoimmune disease where our own antibodies attack our insulin and our beta cells in the pancreas, then we thought, why not try to prevent autoimmunity before it even happens, before we start producing those antibodies against ourselves with propionic acid supplementation in infancy, which is when type 1 diabetes really begins, even though you don't have the symptoms yet. So in the first 18 months of life, this could do 3 things. It could first shift the pH in the stool, which is a way to balance the microbiome. It could preserve the intestinal lining by providing energy substrate for the intestinal cells. And it could maintain the innate immune system active at this critical time when immune training is happening, so that we don't end up recognizing our own proteins as a problem. So to sum up on this idea of the metabolic balance as a potential solution to complex chronic diseases, with metabolomics, we can easily circumvent the caveats of genomics because we can look into the effects of the exposome and the microbiome more directly, so we get this holistic understanding of the disease. And this can also be applied for each subject to focus on their own profile and to enable precision medicine, of course. This can help us to detect diseases earlier because we're not waiting for the symptoms, but we're actually waiting for the first signs of imbalance that will later on lead to disease. We can envision original treatment options, as I showed in the last 2 examples. And we can also deploy adaptive prevention measures for people who haven't developed the disease yet to make sure that we stop this tide rising of complex chronic diseases. And all of this can be monitored with metabolomics because in the end, the metabolite levels is really what triggers the beginning of these diseases. So how do we measure metabolic imbalance? Of course, at biocrates, we work on metabolomics and we're very interested in these diseases. So our latest kit was actually designed to address this type of question. We have metabolites that are related to inflammation, to insulin resistance, to mitochondrial dysfunction, like all those things that are really at the heart and at the core of complex diseases. And if you look at the coverage of the kit, I colored here in red the metabolites that we discuss in the white paper. So in one dot is a single metabolite, and then a box is a group of usually lipids. So you have basically hundreds of metabolites that are absolutely relevant to the study of complex diseases that are covered by this kit. And then in addition to that, you can make clever combinations of metabolites. For example, on the bottom right, the ratio of secondary to primary bile acids that is highly relevant to all the diseases that I've discussed today. Also different ratios of lipids that are measured in the kit to get additional insights into what's happening in your experiment. So I can only encourage you to check out this kit if you want to immediately apply this knowledge to your research. And of course, I want to encourage you to go to the white paper page on our website and download the white paper if this is of interest to you. And if you want to learn even more about metabolomics, I would encourage you to click this learning button that is there, where you find the metabolites of the month and all sorts of very interesting contents on metabolomics. So thank you very much for your attention. And if you want to ask me questions now, please do. And if you want to ask me anything or learn more about metabolomics, you can follow me on LinkedIn. And I post a lot. I will post a lot about the white paper there. And I post a lot also on metabolomics regularly. Thank you very much for your attention.